Cell division, the process by which one cell becomes two identical daughter cells. So let's start with the question of why are cells small? Well, we also need to ask, are the cells of an elephant the same size as the cells of a mouse? And I hope you answered yes, but why? There's two main reasons that a cell divides, so that it does not get too big. Because the larger it gets, the more demands on its DNA. So if we were to compare it to a town and think of a library, and there's a certain amount of books in the library, if the town grows, the population grows, there's still only that amount of books. The DNA doesn't, we don't get more of it because the cell's getting bigger. So to regulate um, what everything in the cell, we would actually need more DNA as the cell gets bigger. So it needs to stay the same size. And the cell would have a lot more trouble moving nutrients and waste across the cell membrane because what happens is the volume of the cell gets much bigger, but the surface area of the cell membrane does not. So the ratio of surface area to the volume decreases, and the cell would have a lot of trouble getting the nutrients that it needs into the cell and its waste out of the cell. So let's do a little background information. The cells of every organism have a species-specific number of chromosomes. Humans have 46 chromosomes and fruit flies have 8. So every organism that we look at, depending on species, will have a set number of chromosomes. Remember that chromosomes are made up of DNA, which carries the genetic code, and it's not visible until it condenses into chromatids. So if we take a look at this diagram above and we look at what a chromatid is, it's one of two sister parts of condensed chromosomes. So here actually this coiled structure would be the chromosome, and it's going to condense and we end up with chromatids. And this is what we typically represent at the top of the screen, what a chromosome looks like, but it's actually two sister chromatids, and they're going to be joined together by what's called a centromere. So you can look up at this diagram. We have two sister chromatids. Okay, each one of these big purple lines is a chromatid, which is condensed chromosomes, and the centromere joins them. Now we can look at the cell cycle. And the majority of the cell cycle is actually what's called interphase. It's the beginning. And this is how the cell is going to maintain its size and be able to carry out all the functions that it needs to be able to do. So the cell cycle is actually a series of events that the cell is going to go through. It's going to grow and then it's going to divide. So during interphase, the cell is actually going to prepare for division. And to do that, we can look at each of these different phases. We're going to look at the G1, S, and G2 phase, and then we're going to look at mitosis. And we can see along this cycle that there's actually checkpoints. So the cell is going to check for DNA damage at two places here, or three places, and then it's going to make sure that all of the DNA is actually replicated. Everything's going through. And we'll look at how the cell does that as well. So let's look at the cell cycle a little closer. G0 would be, the cell isn't dividing yet. But when it's getting ready to, it goes into the G1 phase of interphase. And I want you to notice that this whole thing here, G1, S, and G2, is the time it takes for interphase to happen. And then mitosis is going to take up just this little bit of space here. So at the beginning, the G1 phase, the cellular contents are going to be duplicated. Then in the S phase, the chromosomes are duplicated. And then in the G2 phase, the cell is going to double check the duplicated chromosomes for any errors and make any needed repairs. Then the cell can finally go into mitosis. So the stages of mitosis kind of look like this, and we have our DNA replication. This would actually be interphase, and then we go into mitosis, and we end up with two diploid cells at the end. So let's look at some vocabulary. Diploid, or 2N, are cells that have both sets of chromosomes. So recall that you get a set of chromosomes from your mom and a set of chromosomes from your dad. If we were only talking about one of those sets, we would say the cell is haploid, and it only has one set of chromosomes. But right now we're talking about diploid cells, which will end up with a full set, or both sets. So the first stage of mitosis is prophase. It's the first and longest phase of mitosis, and chromosomes are going to become visible. So that means that our chromatids are actually becoming visible. The centrioles are going to separate, and remember that centrioles are only in animal cells, and they're going to take positions on the opposite sides of the nucleus. The plant cells don't have centrioles, but they're still able to form a spindle like the animal cells do. So the spindle will form, and the chromosomes then become attached to the spindle near the centromere of each chromatid. And the chromosomes actually even coil even more at this point. And the nucleolus is going to disappear, and so is the nuclear envelope. Now we go to metaphase. Think of meta as middle. The chromosomes are actually going to line up across the center of the cell, and each chromosome is going to be attached to the spindle at the centromere. So if these are our sister chromatids, and we can see our centrioles here, our spindle is forming, and then at the centromere we have the spindle attached, and this would be the center plane or the metaphase plate of the cell. The next phase is anaphase, where the centromeres that join the chromatids actually split so that the sister chromatids can separate and become individual chromosomes. So we can see that here. And the chromosomes then start to move to opposite sides of the cell. This would be our cartoon rendition, and this is actually under a microscope in a plant cell. So you can see that the chromosomes are actually being pulled to opposite poles of the cell. Then telophase is actually the last stage of mitosis. The chromosomes unwind, so they start becoming, they look like that loose material again. The nuclear envelope reforms around each cluster of chromosomes, and the spindle breaks apart. The nucleolus then becomes visible in each daughter nucleus. But we have one more stage that we have to look at because we have two nuclei and they're still attached. Well, cytokinesis actually happens at the same time as telophase. Now, think of cytoplasm. It is the separation of the cytoplasm. In animal cells, the cell membrane is actually going to be drawn inward until the cytoplasm is pinched into two nearly equal parts, and this process is called cleavage. Plant cells, the same thing happens, but we have the formation of what's called the cell plate. The cell plate's going to form midway between the divided nuclei, okay, or in between the two new nuclei that are formed. And then the cell plate will gradually develop into a separating membrane, and then the cell wall begins to appear in the cell plate. So if we look at it, this is actually, real cells. We see prophase, we have all that material, we're starting to get chromosomes condensed. Then we have here, um, we're looking actually down here, we're looking at metaphase. They're all lined up in the middle. Then anaphase, the chromosomes are being pulled apart. And here's telophase, where they're still attached, but we have two new nuclei forming. Now we can talk about what regulates a cell cycle, because if all the cells just started dividing uncontrollably, then the systems wouldn't be able to be regulated and controlled. So there's these proteins that are called cyclins, and they actually regulate the timing of the cell cycle in eukaryotic cells. There's two types of proteins that do this: internal regulators and external regulators. The internal regulators are proteins that respond to events inside the cell, and they allow the cell to proceed only after certain processes have happened inside the cell. So for instance, those checkpoints that we looked at before, when all the chromosomes have been replicated to make sure that every chromosome is in place for anaphase and is attached to the spindle so that they'll separate correctly. Those would be internal regulators that control that. Then there's external regulators, which are proteins that respond to events outside the cell, and these direct the cells to speed up or slow down the cell cycle. Things like growth factors. If you have a wound, a cut, you need cells. Say just a paper cut even, you need skin cells to form so that they can cover the wound and heal. So wound healing would be something that would speed up cell division. Embryonic development, you're talking about one cell dividing and becoming many cells to become an individual. And then there's also molecules that are found on surfaces of neighboring cells that often slow down the effect. So they prevent excessive growth so that the cells don't disrupt each other. Because if they just kept dividing, then the other cells wouldn't be able to carry out their functions as well. Sometimes there's problems with these cell regulators, and we end up with uncontrolled cell growth. What happens there is cells don't respond to the signals that regulate cell division, and the cells are going to divide uncontrollably and form masses called tumors. There's many different causes of cancer. Some believe that, smoking tobacco or exposure to certain chemicals, um, things like um, radiation exposure, viral infection, and even genetics. Um, they're looking at the P53 gene, which actually normally stops cell cycle until all chromosomes have been replicated, but sometimes it's defective, like what we're looking at here, and it's not going to tell the cell to stop. So what happens is these cells end up proliferating, or they keep growing. We end up with lots of them. And then you end up with masses in places that there shouldn't be, and it takes away from the body being able to function because it's taking either the nutrients or it's taking up space for processes to occur. And then we have stem cells, which are actually unspecialized cells that have the potential to differentiate or to become specialized in structure and function. And at one time, embryonic stem cells were the only stem cells that were being looked at because those cells, if you think about it, an embryo starts out as an egg and a sperm and they join and start dividing. Well, those cells then become blood cells and cardiac cells and nerve cells, liver cells, all sorts of cells of the body. And that's what they're showing you in this diagram. So scientists have taken that technology and they're starting to say, well, if those cells will divide and become specialized into, what we need them to be, then that would be amazing to be able to use that because we know that they're going to divide by this process of mitosis and be identical after they take on that function. So they're looking at more and more ways in order to use stem cells to help heal people with different cancers or failure of organs, so that they're able to live a better life. So just to review, mitosis is part of the cell cycle. The cell cycle is going to be a process that is going to allow cells to replicate their chromosomes and divide so that they don't become too big and can't carry out their functions. Interphase is the first part of cell division, and it's divided into three parts, G1, S, and G2. In that period, the cell is actually growing, replicating its contents, then replicating DNA, and then prepares for mitosis. And along there, there's checkpoints with those regulators to make sure that everything is done right. And then the cell's going to go through the M phase, or mitosis, so that it's going to go through prophase, metaphase, anaphase, and telophase. And during telophase, cytokinesis, or the splitting of the cytoplasm, occurs. And a little way to remember that is IPMAT, I-P-M-A-T. So if you can remember IPMAT, you can remember the stages of the cell cycle: interphase, prophase, metaphase, anaphase, and telophase.